Hello everyone, my name is Yara and I am a PhD candidate here at Northwestern University, where I study drug resistance in prostate cancer. Prostate cancer is the #1 cancer risk in men, and it's the second leading cause of cancer-related deaths in the United States. This is in part due to drug resistance. This means that even though initially the patients respond to therapy, eventually most patients with advanced disease will acquire resistance and will no longer respond to treatment. Unfortunately, these patients are left with no therapeutic options. Our lab is trying to understand what makes prostate cancer cells resistant to therapy, because we think if we can understand these mechanisms, this will bring us one step closer into finding new treatments. But before I talk about drug resistance, let me explain you how current treatments for prostate cancer work. And in order to do this, we need to understand a little bit about the biology of prostate cancer itself. Prostate cancer is a hormone-driven type of cancer. This means that just as cars need fuel to start and to run, prostate cancer cells need hormones in order to grow and divide. But it is not any type of hormone. It's a specific type of hormone called androgen. In fact, prostate cancer cells are extremely dependent on androgen for growth and proliferation. This means that if we deprive these cells from androgen, we can actually kill the tumor cells. And this can be done through two different ways. It can be done through castration, in which case we are taking away the main source of androgen, which are the testes, and it can also be done through hormonal therapy, in which case we're using drugs to inhibit the ability of the cells to use or to take the androgen. Let me explain you how this works. Imagine we have a cell. In order to work, androgen needs to go all the way from outside of the cell into the cell nucleus. But androgen cannot go there by itself. It needs something to transport it there. And this is the role of the androgen receptor, or AR. Enzalutamide, it's a drug that is widely used for the treatment of prostate cancer, and it binds to the androgen receptor at the same site as androgen does. So you can see how this is a problem. Two molecules are competing to bind the androgen receptor. When enzalutamide wins and binds to the receptor, well, androgen can no longer bind, and therefore it can no longer go inside of the cell nucleus, and it cannot do its job. But as I told you before, even though these treatments work very well initially for patients, resistance is really a big problem. But how do cells acquire resistance to therapy? Well, I like to think of drug resistance as a battlefield, where only the strongest survive. If we take this analogy further, let's imagine that we are trying to fight against the prostate cancer cells army, and that this army is heterogeneous. This means that it's composed of different types of warriors. Maybe we have one warrior with a sword, one with a shield, one with an axe, one with a mace. And let's also imagine that we've been very effective and that we've managed to kill mostly all of our warriors, except for one type. If we wanted to kill also this type of warrior, it will be very useful if we knew which type of tool is this warrior using. In a similar way, prostate cancer cells will activate different genes in order to become resistant to therapy. The challenge is to identify which genes are being activated by these prostate cancer cells. And this is not an easy task, because our genome contains about 20,000 genes. So luckily for us and for me, we now have CRISPR. And with CRISPR, we can activate every single gene in the genome. And therefore, now we can study the contribution of every gene in drug resistance. Let me explain you a little bit more about this concept. So let's imagine we have sensitive cells. This means that these cells die with the drug. They are not resistant. And we take CRISPR to activate different genes. Then we can treat these cells with our drug of interest, in this case, enzalutamide. And at this step, most of the cells will die. But some cells will survive. The surviving cells have activated genes that are very important for resistance. And using sequencing technologies, we can actually identify which are these genes. And this is very important because if we want to win the battle, we really need to know our enemy. And this is not only true for our warriors analogy, but it's also true for our prostate cancer cells. Because by knowing which genes are being activated, then we can plan a better strategy to silence these genes or the effect of these genes using drugs. And if we take this one step further, we can actually think of patients that are being assessed to see if these genes are being activated after they obtain resistance or acquire resistance. And then with this information, we can use drugs that target specifically these alterations. Trying to overcome drug resistance in prostate cancer, it's not an easy task. And identifying the genes that are important for prostate cancer resistance is only the very first step. Many, many years could go by until we can actually use this knowledge and transfer it into the clinic, or it can be actually used by a patient. But as every journey, this one as well begins with a single step. Thank you.